Xtalks is the industry leader in connecting life science, food, healthcare, and medical device professionals. The Xtalks Life Science podcast features insightful conversations with industry experts about all things pharma, biotech, and more.
Hello and welcome to the Xtalks Life Science podcast. I'm Ayesha, Senior Life Science Journalist at Xtalks.com, and this week I'm delighted to introduce our special guest on the show. We have with us today Dr. Lawrence Blatt, who is the Chairman and CEO of Aligos Therapeutics, a company developing targeted therapies for liver diseases like MASH, as well as viral diseases, to address important unmet medical needs. Prior to co-founding Aligos Therapeutics, Dr. Blatt served as the global head of infectious diseases and vaccines at Janssen Pharmaceutical companies of Johnson & Johnson from 2014 to 2018. He also co-founded several biotech companies, including Alios BioPharma, which was acquired by Janssen in November 2014, and Alveo Technologies. Dr. Blatt has also served on the board of directors of multiple companies as well. Dr. Blatt received an MBA from California State University, Northridge, and a PhD in Public Health Administration from the University of La Verne. In March, Aligos announced the dosing of its first patient in its Phase 2A trial for a novel thyroid hormone receptor beta agonist, ALG 055009, which is designed to address the root fibrosis of MASH. The drug is in the same class as Rezdiffra, which was approved this year as the first treatment for MASH. Aligos also shared positive data at the European Association for the Study of the Liver Congress last year in Milan, Italy, for one of its candidate therapeutics for chronic hepatitis B. My apologies, this year in Milan, Italy. So to learn more about the work Dr. Blatt is leading at Aligos Therapeutics in chronic liver disease and viral diseases, let's turn to him now. Thank you so much for being on the show, Dr. Blatt.
Well, thank you for inviting us. We have a lot to talk about today, so we should probably just jump into it.
Absolutely. So, Dr. Blatt, I'd like to start off by asking you more about what led you to co-found Aligos and what the company's key missions and goals are.
Well, you know, a lot of the major breakthroughs that happen in biotechnology and pharmaceuticals occur in small companies. As you mentioned, I've been a founder of several small companies that have gone on to ultimately be acquired by large pharmaceutical companies. We were doing great work at Janssen in the infectious disease group, but I felt in 2018 that it was time for me to get back into a small environment and target therapies using a small team of scientists. So in 2018, I resigned my position at Janssen and formed Aligos Therapeutics. And in fact, a number of scientists who were working with me at Janssen wanted to come along and join me on my new team. And those were scientists that actually were part of Alios BioPharma previously. So it's a group of scientists that have worked together for many years. And we have a great team and great camaraderie and dedication really to creating novel therapies for patients.
And currently, what are some of the unmet needs in liver diseases like MASH? And how does Aligos' mission align with these current unmet needs in terms of the treatment of the disease?
So liver disease, interestingly enough, can be characterized as an insult to the liver, and that may come from a metabolic defect, as is the case in MASH, or a viral infection, as is the case for hepatitis C and hepatitis B. And the interesting thing about those two diseases, they start with very different insults, but they end up in the same place. So the insult causes an inflammation of the liver and a damage to hepatocytes or liver tissue. That damage results in a wound healing response, which deposits, instead of normal hepatocytes, scar tissue in the liver. We call that fibrosis and cirrhosis. And whether you're suffering from MASH or HBV or HCV, you can end up with end-stage liver disease, which means your liver has failed and you could no longer metabolize toxins. There's a number of other comorbidities that occur when that happens. And you can go on to liver cancer, or what we call hepatocellular carcinoma. So both MASH and HBV cause these downstream clinical sequelae and ultimately result in the same ending for the patient.
And can you tell us about Aligos' developmental pipeline and the company's approach to really developing improved liver disease treatment, since this is an area that, as you've mentioned, there's a huge unmet need, and we've only seen the very first treatment for MASH this year. So how is Aligos really trying to meet those unmet needs through its developmental pipelines?
So for MASH, we started our program, our discovery program, and LG-009 was invented in-house by our scientists. And what we did was we surveyed the field of what was then called NASH, non-alcoholic steatohepatitis, and that name was changed to affirm what the disease is, not what the disease isn't, really led by the AASLD president, Nora Thoreau, which is the Metabolic Associated Steatohepatitis. That's where the name comes from, or Metabolic Dysfunction-Associated Steatohepatitis. And what we found was there were a number of companies that were pursuing molecules for MASH that in phase 2 had poor data, but they went on to phase 3, and of course those trials failed. Whereas we saw the data from what ultimately became Rezdiffra, which was a first generation beta thyroid agonist. So the thyroid hormone receptor has two isoforms, the alpha and beta isoforms. And the beta isoforms primarily found on the liver. And so they were developing a novel therapy with a rather old drug, actually. And they were getting pretty good phase 2 data. We did a survey of that compound. We found that there was a lot of pharmacological deficits of the compound. So the exposure or pharmacokinetics were not optimized. The drug has about a 3-hour half-life. It also has hypervariable PK. So among different individuals receiving the same dose, they don't really get the same exposure. And it has what we call nonlinear PK. When you give a dose of 80 milligrams and advance to 100 milligrams, you actually get 3 times the exposure of the drug. So it has nonlinear, hypervariable, and a short half-life. And we saw that as an opportunity. In addition, the drug is not very potent. It has about a 2 micromolar potency compared to modern drugs which have nanomolar potency. And we can see that difference in our dose. So we're dosing 0.3 micrograms compared to 180 and 100 micrograms. So 0.3 to 0.9 micrograms is our dose compared to nearly milligram doses for the Rezdiffra. So that's really the unmet medical need there. And the bottom line is, because these patients don't have exposure that's optimized, some patients do well. The patients get the optimal dose, do well. Some patients are underexposed and they don't get the benefit of the drug. And so we see this as an opportunity to bring the overall efficacy up on the Rezdiffra data using our drug 009. Now, what's very important, I should add here, that the liver is a regenerative organ. So our liver cells are turning over and new hepatocytes are coming into the liver. That's very different. If you scar your heart or your lungs, you're scarred for life. And what this means is there's the potential to reverse that scar, reverse fibrosis. And one of the amazing data points that was generated with Rezdiffra was not only improvement in the fat deposit or the NASH or MASH score, but also a non-worsening and even improvement in the fibrotic score. So that's a great benefit to patients, because I think it's important to note for MASH, we're not treating these patients to defat the liver per se. We're treating the patients to prevent end-stage liver disease and liver cancer, and that is a result of scarring of the liver. And I always equate it to statins. People say, well, I'm on a statin to lower my cholesterol. And in fact, you're on a statin to prevent the risk of cardiac disease and stroke. And lowering cholesterol is a biomarker on to that endpoint, that clinical endpoint. So it's the same kind of thinking here. If we turn to HBV, it's a similar story. The current therapies that are approved are inadequate. They do a good job of blocking the virus from replicating, but they don't do a good job of arresting the disease completely. We have a novel therapy that we just announced positive data, and we'll get into that, I think, more later. But again, HBV, high unmet medical need, 300 million patients throughout the world. And HBV is, in the world, the leading indication for liver transplant out there.
Wow, definitely a significant unmet needs out there, and it's great though that having identified the targets, and then now you can go after them and know exactly what you need to do. So
right, exactly.
Wonderful. And so could you tell us more about the first dosing of a patient in the phase 2b trial for ALG 055009?
Yeah, so again, 055009 is a beta thyroid agonist, meaning it stimulates the beta isoform of the thyroid hormone receptor. I think we have some questions later about the mechanism and why that is a good thing to do in MASH. But we did enroll this study in 6 weeks, which is really record time. And I think it shows the enthusiasm for new treatments for MASH. And we were able to enroll the trial rapidly, even as Rezdiffra was launching. So I think it shows the unmet medical need and the gap that needs to be filled with the Rezdiffra data.
Mm-hmm. And yes, actually, we should maybe speak about the mechanism of action for sure, of how the compound works. You did mention that it's a thyroid receptor agonist. So could you tell us more about the mechanism of action?
Yeah, so I think, as most people know, thyroid hormone is a master regulator of a number of bioprocesses in the body, including metabolism. The liver in a MASH patient has a specific defect, and that defect is that there's an enzyme in the liver that actually converts the active thyroid hormone T3 into what we call reverse T3. And that means that the liver becomes hypothyroid, so it's not being stimulated in a normal way. And what happens then is the metabolic pathway for fat changes because you're not inducing the normal metabolic pathway around the beta thyroid hormone receptor. And you begin to create what we call reactive species of fat. So these are oxidative reactive species that actually form in the liver and deposit in the liver and are now damaging the liver. And what we believe is going on is you're correcting that metabolic defect in a MASH patient. You're stimulating the beta thyroid hormone receptor and allowing restoring normal metabolism. Now, one of the important points about this beta thyroid hormone receptor field is that at the same time that you stimulate beta isoform, you do not want to stimulate the alpha isoform. So the patients are hypothyroid in the liver, but not throughout their body. So if you overstimulate alpha, then you'll give somebody basically clinical hyperthyroidism, and that could lead to cardiac disease and other downstream effects. So what we've done is we've created a molecule that has the highest ratio of alpha to beta stimulation. So our ratio is around 4-to-1, whereas Rezdiffra is around 2.5-to-1. And that means that we have a better chance of finding the therapeutic index where we optimally stimulate the beta isoform and not the alpha isoform. So that's what we're doing with our molecule.
That's fantastic to have that approach where you're really specifically targeting the isoform of interest. And
yeah,
that's what it's all about, precision and targeted medicine and therapy. So it's really great to learn about.
And this is a difficult task because the difference
I can imagine, yeah.
The alpha isoform and the beta isoform is one amino acid. So we really had to lock in on the structure. We did structural biology to find this molecule through our chemistry group, and really lock in on that structural difference between those two isoforms.
So that you can achieve a good therapeutic index.
That's right. That's right.
Strictly, that's relegated to the liver. So that's really important in terms of drug toxicity and all of those things.
And these patients are going to have to be on this drug for life. So you can't make somebody hyperthyroid for life. That would be a bad outcome for those patients. So it's very important to kind of walk that line between beta stimulation and alpha stimulation.
And I'd love to learn more about Aligos' chronic hepatitis B program. I know that the company recently shared clinical data at the conference in Europe that I mentioned happened about a week or so ago in Milan. So I'd love to learn more about that program.
Yeah, so we presented some very exciting data there. So HPV is a very complicated virus. It has a lot of tricks to maintaining its chronic infection in patients. One thing that HPV does is it deposits in the nucleus of a cell a mini chromosome, which we call cccDNA, or covalently closed circular DNA. In addition to that, HPV integrates a portion of its genome into the chromosome of the host. And primarily what gets integrated is something called HPV surface antigen. And that causes the production of high quantities of soluble surface antigen. And that surface antigen is actually a strong immunosuppressor, which blocks T cell responses and allows the virus to remain in a chronic state. So that's a long-winded way of saying there's a lot to do in HPV. Now, current therapies, they block the replication of the virus or the copying of the virus. Those are nucleoside and nucleotide analogs, but they don't do anything to the cccDNA. So that's the reservoir for the virus. And if you can't affect cccDNA, you'll always maintain this chronic infection. The other thing about current therapies, nucleoside and nucleotide analogs, is they don't fully suppress replication. So there's been experiments where that's been shown there's low level of virus in the blood of these patients. And if you take that virus out of patients, if you extract the blood and take it out and try to infect experimental mice, they can actually get hepatitis B from the blood of patients with low level replication. We have a novel mechanism in HPV. It's a third generation capsid assembly modulator. So this is a drug that binds to something called the HPV core protein, which assembles into the capsid or outer shell of the virus. And by blocking this assembly of the virus, you're actually, one, blocking replication, but two, you're blocking a second step in the virus, and that is the replenishment of cccDNA into the nucleus. So by blocking those two stages, you're affecting multiple parts of the life cycle. What we reported at the meeting was full suppression. So there's two status of HPV patients. There's E positive patients, that's another antigen, the E antigen of HPV, and there's E negative patients. So we reported for the first time data on E negative patients using a very sensitive assay to measure HPV DNA. And with that assay, we showed that 9 out of 10 patients had target undetected. That means they had less than 3 international units of HPV in their blood after receiving our therapy, actually for only 24 weeks. We had that level of suppression. We kept the study going out it's going to go 96 weeks. Right now, those patients are around week 60. What's interesting is our monotherapy is fully suppressing HPV replication with no drug resistance, so we have no breakthrough in that patient population. Now, in the more difficult to treat patients, the HPV E positive patients, we've shown complete suppression of HPV DNA after 48 weeks in 6 out of 10 patients, and by 72 weeks, 9 out of 10 patients. So it takes a little bit longer for them to get fully suppressed. But what's very interesting is we're showing reductions in HPV antigens in these patients. So we see reductions in HPV S antigen, HPV E antigen, and what's called core-related antigen. And that is an indication that we're suppressing cccDNA. And that's not something that nucleoside analogs can do. And so this really puts our drug on a pathway for regulatory approval based first on chronic suppression, so achieving a higher rate than nucleoside analogs can achieve with full suppression of the virus, but also secondary endpoints for reducing these antigens that are so important for the maintenance of the virus. We're going to be moving into phase 2 shortly, sometime next year. And we're going to be doing some pretty unique science here. We're going to be taking paired liver biopsies, and we believe that by fully suppressing the virus with this therapy, we're going to reduce the amount of cccDNA. So we're actually going to measure cccDNA at baseline and cccDNA at 48 weeks. We're also going to measure the amount of virus that is integrated into the chromosome, and we hope to see reductions in that integration. And that's very important because the integration of HPV is a random event. And sometimes HPV can integrate in sites which activates oncogenes. So that's one of the ways that HPV causes liver cancer. So we're going to be looking at not only the primary endpoint of DNA suppression, but multiple secondary endpoints, including antigen reductions, reduction in cccDNA, reduction in integrins, and also reduction in HBV RNA. All of our patients are HBV RNA negative by week 16 of our study, and HBV RNA correlates with liver cancer. So patients with high levels of HBV RNA have a greater risk of going on to liver cancer than patients who have lost HBV RNA. So those are all the different endpoints that we're doing in the study, and we're very excited about the data we've just presented, as well as moving our drug into phase 2. I should say these data are unprecedented. Earlier capsid assembly modulators were unable to achieve the endpoints we're achieving, and the reason for that is we've improved the potency of our capsid assembly modulator compared to others. We have a picomolar potent molecule, so sub-nanomolar potency, and we also worked on the pharmacokinetics of the molecule. So you can see that that's a theme at Aligos. We dial down potency, we make our molecules very potent, and we also improve the pharmacology of the molecules.
Absolutely a great common theme to have across your different programs. And speaking of other viral diseases, I know that Aligos also has a COVID-19 protease inhibitor program, which is actually is a partnership with public government agencies like the NIH. So I'd love to learn more about that as well.
Yeah, so one of the things that we did, and this was based on personal experience of mine, is we've actually selected what we call a coronavirus protease inhibitor, not a COVID protease inhibitor. And what I mean by that was when we did our experiments to find this molecule, we screened against all known strains of coronavirus, including SARS-1, MERS, the Middle Eastern Respiratory Syndrome, as well as SARS-CoV-2. Additionally, we've screened against 2 seasonal coronaviruses. So coronavirus has been with us for quite a long time. And our thinking around this was that, in the past few decades, we've seen 3 coronaviruses jump from animals to humans. And it's not if there's going to be another coronavirus jump, it's only when. And what happens when these new viruses come out, people like to throw existing drugs at them. And by and large, those existing drugs don't work. And that's because antiviral therapies are not broad spectrum. They tend to be therapies made specifically for virus. So, for example, if you took an influenza drug and tried to treat coronavirus, you would not have any inhibitory properties of that drug. So the reason the NIH and NIAID is so excited about our program is that we have a pan-coronavirus molecule, and they're, like us, worried about the next coronavirus pandemic. Now, I mentioned this was a personal quest for myself. I had been involved in SARS-1. So when the initial SARS outbreak came, I was running a clinical trial with a drug called Infectious, which is a engineered type 1 interferon in Toronto, and we actually published positive data in JAMA using interferon for SARS-1. And I always said at that time that if there was ever a coronavirus that was highly infected, we were going to be in big trouble. And then, SARS-2 came along. We can debate the origins of that. It's probably beyond the purview of this interview. But we, at that time, immediately mobilized and began to work, as soon as the virus was identified, began to work on a pan-coronavirus protease inhibitor. And we actually enlisted collaborators at the University KU Leuven, Christian University Leuven. And that's because there's a fantastic virology center there called the Rega Institute. A number of antivirals have come out of the Rega Institute, and I was personal friends with Johan Netz, who is the director of that institute, and we formed a collaboration with them to develop our drug. And the reason is they have a BL4 facility where they can grow all these very highly pathogenic viruses like SARS-CoV-2, MERS, and SARS-1. So our drug was formulated to act against all coronaviruses, and that's really why the NIH is paying attention to it. We just reported data at ECMO on our phase 1 for the drug. It does not require ritonavir boosting. You may know that Paxlovid uses an old HIV drug to inhibit metabolism of that drug, and that causes a lot of problems. not least of which is this horrible metallic taste. I don't know if you ever took Paxlovid, I did, but it causes a horrible metallic taste, and that lasts throughout the dosing of the compound. But the more important thing is that inhibiting cytochrome P450 is detrimental to other drugs that are given alongside. So patients that are at high risk develop bad outcomes for COVID, have many comorbidities, and need to be on drugs like blood pressure drugs, cardiac drugs, statins, and other drugs that are metabolized by the enzyme that the ritonavir is inhibiting. So co-administration with ritonavir actually knocks out a lot of patients who would be eligible, who need antiviral therapy. So we developed our drug to avoid that issue and also be a pan-coronavirus.
It's very interesting how that approach came about, and how, like you said, coronaviruses have been around and will continue to be around. And so that's wonderful that you also have a program that's targeting those. And so I'd like to actually, in the beginning, you mentioned that you, of course, moved from Janssen and then co-founded Aligos, and how you wanted to get back into the smaller biotech, mid-biotech space. And you brought, or some people from Janssen came over as part of your team. And so I'd like to kind of know like what the differences are between working at a small, mid-sized biotech and the larger one, and what really are some of the advantages, and why that makes, or perhaps in the landscape of drug development today, what are the sort of the pros and cons of each?
So again, if you look, just across our industry, most of the major breakthroughs that occur in medicine start out in small biotech companies. And I think the real reason for that is really focus and determination. So in a large pharmaceutical environment, you have many factors that are going on, including, revenues, which are driving the amount of R&D that you can spend. And I always say that big pharma, really runs off a profit and loss statement, and they have an obligation to pay dividends on their stock, and that tells you how much they can spend on R&D. And there are often external things that are happening that can impact an R&D organization. So one drug going off patent in an indication not even related to what you might be working on is going to affect the R&D spend, because that affects the P&L statement, which affects the dividends. And so determination and really focus are really hampered in a large environment. Now, that's not to say that big pharma isn't important. I think once you get into phase 3 and once you get into distribution and marketing, there's no better way to do it than big pharma. But because of all these distractions that occur in a large company, it's really hard to focus in and innovate, because it's usually the first things that go in a lean year are the things that are not as advanced in a big pharma. So I really focus my career in discovery. I've been involved in the discovery of many drugs that are marketed drugs to date. And that is really best done in a small environment. So we have a team of around 60 people, and we've accomplished quite a lot. We've put several drugs in the clinic, several of which have been invented in-house. Our HPV CAM was co-invented with researchers at Emory University, led by Ray Shinazi, who Dr. Shinazi not only is a personal close friend, but has been involved in many breakthroughs in antivirals, including Sovaldi for HCV, as well as a number of HIV medications. So we collaborated with Dr. Shinazi's lab, and 184 is the result of that collaboration.
Thank you for those insights. And definitely, I think small biotech is kind of where it's at. And we've seen that during COVID too. I mean, the vaccines that were developed, was big pharma came in to partner with them, but essentially they were at the smaller, mid-sized companies that were driving the discovery for sure.
Yeah, and I'll tell you something interesting about COVID. When COVID hit, interesting story. My mother called me up and said, "Well, what do you think, about 2 weeks and everything will be back to normal?" And No, it's going to be 2 years. And what we did at our company, we actually did not shut down our company during COVID. We instituted policies. We were doing mask wearing right at the beginning, well before the NIH and CDC mandated it. We had a temperature gauge that measured people's temperature as they walked in the office early on. And of course, if you didn't feel well, you didn't come in. And we did not have any, internal outbreaks of COVID. And we actually worked through the entire time and were able to be very productive in that time period.
Yep. I think it's about, taking the appropriate measures at the appropriate times, for sure. Yeah. As we've learned. And so what key milestones can we expect from Aligos looking forward in the coming months, in the coming years? What are you most looking forward to?
So the coming months, the study you mentioned for our beta thyroid agonist, ALG-009, we call it the Herald study. That reads out in the fourth quarter of this year. And what's important about that study is it's measuring an important clinical surrogate endpoint. It measures fat in the liver by MRI testing, and that outcome has been correlated with improvement in liver histology. So we're hoping and betting, based on our chemistry and our improved PK and potency, that we're going to have a better result on MRI-PDFF compared to Rezdiffra, and that's going to really set us on the path for a best-in-class drug. I like to think about it in this way. Yes, we are behind Rezdiffra, but if you look at a chronic drug like the statin market, for example, the leading statin, Lipitor, was launched fifth to market in 10 years after the first statin was introduced. So there's time for improved drugs in these marketplaces. I think it's very important that to know that another important factor here is there are very few mechanisms for MASH that are oral drugs. A lot of the other drugs are injectable, and think about having to inject something for the rest of your life. That's a difficult process. So we like to focus on our oral therapies in our company. So in addition to the MASH output, we're going to be actually hosting a key opinion leader event for HBV. We're coming up in July. We haven't announced the date yet, but we will be going through our data with a number of KOLs and talking about our regulatory strategy. We currently have submissions into the FDA to talk about our phase 2 study. We hope to get feedback by that time and give data on that. So that'll be quite important. And then we are working on external funding, government funding, and actually private funding around our COVID drug to move it forward, or our coronavirus protease inhibitor. So we'll be announcing data on that. So a lot happening in the company, a lot of milestones coming up. And it looks to be that, we have a number of best-in-class drugs for our portfolio, as well as, in the case of 184 for HBV, first-in-class drug. So that's moving forward. We do have a strong research pipeline, so we're not stopping at these molecules. We have a number of things we're looking at preclinically right now, and we'll be announcing more about that in the coming months.
Well, we'll be sure to keep our eyes peeled for all of those milestones, and we'll definitely be following Aligos here at Xtalks for the wonderful and exciting work that's happening at the company that you're leading.
Well, thank you so much, and it's been a pleasure to talk to you today, and happy to answer more questions in the future should you have any.
Likewise, we look forward again to speaking with you again to learn more about the exciting work that's happening at Aligos. So, Dr. Blatt, thank you so much for your time today and for your insights.
Thank you.
All right, that's the end of this episode of the Xtalks Life Science Podcast. If you liked today's show, don't forget to rate, review, and subscribe. Thank you, everyone, and we shall see you next week. Thank you. Bye for now.
To join the Xtalks community, visit xtalks.com and register for exclusive access to curated content like live webinars, news articles, and educational videos. The views and opinions expressed in this podcast are those of the speakers sharing them and should not be taken as professional advice, nor do they necessarily reflect the policy or position of Honeycomb Worldwide. For further information, email us at podcast@xtalks.com.